developments that really take high dimensional data and try to come up with interpretable insights from those are going to play an increasing role in studying cancer and other diseases.
In this episode, CancerNetwork spoke with Dr. Smita Krishnaswamy of the Yale Center for Biomedical Data Science and Yale Cancer Center about findings from a study published in Cancer Discovery. Therein, an AI tool, the Archetypal Analysis Network, or AAnet, aided in characterizing the diversity of individual tumor cells in triple-negative breast cancer. The conversation began with an outline of the tool and how it was applied to triple-negative breast cancer. Then, Krishnaswamy outlined key findings from the manuscript, including an emergence of 5 significant archetypal cell states and a heterogeneity between primary tumors and metastatic sites. Krishnaswamy then highlighted practical requirements for applying AAnet to clinical samples, the ability of AAnet to stratify other heterogeneous cancers, and additional developments in biomedical science data that might impact the cancer treatment paradigm.
Archetypal Analysis Network is a neural network that we developed. Basically, what it does is it computes a different representation of the data. So it takes the high dimensional single cell data that, for example, in this case, we measured on triple negative breast cancer, and it comes up with a low dimensional simplex form. And this is a regular simplex or a K-gon where the corners represent the archetypal cells or sort of the more specialist extreme cell types. And the other cells can be described as a convex mixture of these archetypal cell types. So this gives you a automatic mixture representation of the data in a low dimensional latent space that you get by nonlinearly transforming the data. So you can go into this space and out of this space. And we've used this to understand what the cellular heterogeneity in triple negative breast cancer mouse model is, both in primary and metastatic tumors. So what we found is that when you look at the primary tumor, we found about 5 significant archetypal states. So we embedded it into a 5-gon. And these states had distinct characterizations spatially as well as metabolically. So they seemed to be, something that's very real. Like if you go to the corners, they have different spatial configurations and different metabolic pathways, as well as gene expression signatures associated with them. What was also interesting was we looked at different metastatic sites. We looked at lung, liver, and lymph node. And these metastatic sites also, for the most part, had the very same archetypes. One of the metastatic sites, the lung didn't have, for example, the hypoxic one, and one of them had an extra one, but for the most part, it seemed like the same heterogeneity was recreated in the tumor, in the primary tumor as the metastasis. We also saw that we could use some of these signatures to treat or try to ablate some of the cell types, which resulted in lower incidence of the metastatic tumor. So we looked at the metabolic signature of the hypoxic archetype, and we saw that it was using glycolysis, the glycolysis pathway a lot, and we saw that knocking down GLUT3 lowered the occurrence of this kind of metastasis again in the mouse model. So those were some of the key findings from this particular paper. If you have discrete clusters, so sometimes people cluster data that's continuous, that would just be kind of making sort of a semi-arbitrary split in the data. But if you don't actually have cluster structured data where it's separated, one thing that's interesting is that the cells lie along a continuum, and that's why these are extremal states that we pointed out, and there are several cells that are somewhere in between these states. It could also imply that these states can transition from one to another. One of our hypotheses is that the proliferative archetype may be the archetype that sort of seeds the metastasis, and then the other archetypes are created from there. And it also shows that also the fact that you recreate the same heterogeneity may show that you kind of need these different cell types to kind of sustain the tumor. And so, like we did, ablating some of the key archetypes might be useful in deriving cancer therapies. We showed how to apply it to some human clinical samples that we found of triple negative breast cancer. So you can, if you have single cell data, you can basically do the same thing. You can, if you, let's say, have a tumor biopsy and you measure it in single cell, you could actually measure the heterogeneity and the different cell states, and you can potentially look at the signatures of these cell states. And I guess if you don't necessarily have single cell data and you have some kind of bulk data, you could try to project it to into the continuum. But I think in the future, as single cell technology becomes more and more prevalent, I can envision a future where tumors are measured in single cell because there is single cell level heterogeneity in the tumors. And a couple of other things that we're sort of studying is how treatment like chemotherapy affects the heterogeneity and what cell types sort of come out of it that lead to the resistance. So I think there's sort of a variety of ways you can use it as long as you have the appropriate data type, which is single cell sequencing. We've already applied AAnet ourselves uh, to an application in pancreatic cancer with the lab of Mandar Mazumdar here at Yale. That manuscript's under review. I think there's a preprint online. There, the idea is we're looking at the different cell types that are prevalent in obesity-driven pancreatic cancer. And we're also looking at dynamically how these archetypes change over time to give rise to some of the cells that are most responsible for progression of this kind of cancer. I think that you could probably look at any cancer and see what the salient cell types are, potentially look to targeting all of them or the ones that are most likely to be that have sort of a metastatic or EMT-like signature, things like that. So I think you could apply it to any cancer. One thing that we've really been involved in, also with the lab of Dr. Christine Schaefer, as well as with the other lab that I mentioned, is looking at the dynamics of these cell types. So how as the cancer progresses, maybe initial cell types progress into later cell types, and we do that using what are called neural network flows. So these are neural networks that learn continuous progression from static snapshot data. And in this cancer, we have a um, in vitro model that we looked at over 5 different time points. We looked at how single cells that are seeded in matrigel over time can grow into tumor sphere formation. And we're looking at how the dynamics in the gene expression and the network that kind of drives these dynamics might be targeted. So basically, these dynamics learning neural networks that we sort of pioneered with TrajectoryNet, and then we had a method called MioFlow, and then later some of our students and others have other networks called flow matching. So these basically take the single cell population data and project it to flows to show how these develop. I think these kinds of things as they progress could have a big impact on not only sort of which cells start the cancer most prominently that then lead on to the metastasis, but also uncovering the underlying networks. I think that deep neural networks and other kinds of AI are very important in these developments, but I think it's very important to design them or redesign them to understand sort of the geometry and topology of the cellular state spaces in the tumor and work with highly unlabeled data. So at the moment, they're really designed more for natural language production, maybe image classification, but I think that these developments that really take high dimensional data and try to come up with interpretable insights from those are going to play an increasing role in studying cancer and other diseases.